Hello everyone, welcome back to Bio tutorials. In this tutorial, I will talk about restriction enzymes and how you can choose your restriction enzyme using NEBCutter online tool. First, let's talk about what is restriction enzyme. A restriction enzyme is a protein isolated from bacteria that cleaves DNA sequences at sequence-specific sites, producing DNA fragments with a known sequence at each end. The use of restriction enzymes is critical to certain laboratory methods, including recombinant DNA technology and genetic engineering. Discovery of restriction enzymes. Restriction enzymes discovered in a bacteria as a defense system against viruses. As you can see in this picture, restriction enzymes attack the DNA of the virus and try to make degradation to it as a defense response. Structure of restriction enzymes. Restriction enzymes are typically composed of two subunits forming a dimer, as you can see here, a subunit and another subunit forming a dimer. Each subunit has an active site that interact with DNA. Enzymes' active site recognizes specific DNA sequences, usually from 4 to 8 base pairs long. What's the function of restriction enzymes? Restriction enzymes bind to DNA at specific recognition site and cleave the DNA backbone, often creating sticky or blunt ends. As you can see here, this is a sticky end or a blunt end. Sticky ends overhanging single-stranded ends. You can see here the overhang. Blunt ends, straight cuts with no overhangs. As you can see here, it's a straight cut. Biological role: protect bacteria from viral infections by degrading foreign DNA. Applications of restriction enzymes. We are talking about the concept of the industrial use of restriction enzymes. We are using restriction enzymes in cloning, used to insert genes into plasmids for recombinant DNA, as you can see in this image, or DNA mapping, generate DNA fragments for creating restriction maps, or for genetic engineering, modify genetic materials in organisms, or in diagnostics to identify genetic mutations or variations by techniques like RefLab. Now let's go to the practical part using NEBCutter 3.0 online tool. As you can see, this is the interface of NEBCutter 3.0 online tool to choose your specific restriction enzyme. All you are going to do is to paste your sequence of interest here and check if it is a linear or circular sequence and do submitting. If we assume that we are looking for restriction enzymes for CYP2C19 messenger RNA, we would just go to the FASTA file of our sequence and copy and paste our FASTA file. We just copy the sequence here and paste it in the input here. Our sequence is not circular, so I will not check this option. Name of the project is not important, and I will just submit. And finally, we are here with the result page. As you can see, it is divided into categories, and down there you will see the length of your sequence and if it is linear or circular. GC content in our case is 41%, ET content is 59%. In the first category, the graphical view, it shows you your sequence including the open reading frames. As you can see, we have two open reading frames, one was 490 amino acids and the other is 116 amino acid. The software puts each restriction enzyme in the position it will cut in your sequence. As you can see here from the enzyme list, it's a list of all the available restriction enzyme, and it said here in number of cuts about how many times a specific restriction enzyme will cut in your sequence. As you can see, this restriction enzyme will not cut in your sequence. Another example, ACIL will cut 4 times in your sequence, and so on. The third tab is your sequence, is your original sequence. And here in the ORF summary, as we said, we have two open reading frames, one with 116 amino acid and the other with 490 amino acid. So let's go back to the graphical view. In the graphical view, you can choose your enzymes. For example, if we wanted to pick up restriction enzymes that cut our open reading frame, this one, the 490 amino acid open reading frame, ORF, we can choose a restriction enzyme that cut at the end of the open reading frame and another that cut at the end of the open reading frame. By the same method, if you are going to pick up a restriction enzyme from a published paper, you can make a check that the restriction enzyme is suitable for your research by doing this research on the NEBCutter 3.0 online tool. We have reached the end of our video and see you in another tutorial. Thanks.